I guess what would you say, Dr. Macauley, that you are really into glyco? You're the glyco guy, pretty much, right? Isn't that?
Yeah, so that means carbohydrates. That forms the basis of our research. I'm not going to talk about that so much today, but that's kind of a central theme in our research.
Yes, absolutely. So he's a biochemist and researcher. Really is a testament to the potential of harnessing the body's immune system to fight Alzheimer's disease, which is why we're all here. By focusing on the protein called CD33, found in the brain's immune cells, his work aims to enhance the brain's natural defenses against neurodegenerative plaques. And this research not only opens doors to new therapeutic strategies, but also offers a beacon of hope for slowing down or even preventing Alzheimer's disease in the future. And so with that.
Okay, thanks, Holly. My lab, say hi to the people at the back. They're going to be the ones that are going to be touring you later. We're scientists first and foremost, but scientists sort of define themselves in different ways. I've got this really pretty picture, and there's 3 colors on here. I'm going to tell you what they are. So this is a picture of a brain. It's not a human brain, it's a model brain that we use for mice. And the yellow here are the neurodegenerative plaques. So this is the first pathology that happens in Alzheimer's patients 10 to 20 years before we see actual disease. So they're really the thing that kick off the disease. It's interesting, some people that get plaques never go on to get Alzheimer's disease. And so why is that? Well, scientists now think that it's the response of the immune cells to that. You can have kind of good immune responses and bad immune responses. And so in red, the second color here, these are all the immune cells in the brain. Okay, so these are called the microglia. And you see that some of these really well-developed plaques, the immune cells are basically all around. It's almost like a fence, if you will. That's how I like to think about it. It's a little bit more active than a fence, and we'll get into that. The blue, by the way, is the nuclei. Okay, so this allows us to count 1, 2, 3, 4, the number of microglia. The other 2 are sort of like accessory labs and have specialized equipment.
Hope you guys are enjoying your tour so far. So this is a flow cytometer, and basically what it does is it allows us to detect individual cells and what they have on their surface. So I've pulled up some data here that someone in our lab has previously collected, and it just looks like a bunch of colorful colors, but to us we're able then to separate various populations and ultimately see expression levels, if that makes sense. So these are microglia from our mouse that express CD33, and so they have a marker that we can see on the flow cytometer that allows us to have this expression level here, knowing that our microglia in those mouse are effectively expressing that protein, which is very cool. So I've pulled up a quick little video because it does a better job at explaining what's happening with the machine. If you want, you can come here. And kind of explains what's going on. So basically here they're just showing a population of cells that have been labeled. So that's these little colorful dots there.
So you've got different types of cells that are labeled there.
Yeah.
So it looks like neutrophils there, and those are leukocytes, just plain. And then what are those ones?
And based off of like how they reflect light, you're able to then separate different cells from. So we can take like a blood sample, for example, and pass it through and be able to selectively look at one population of cells compared to another.
So these are kind of what the microglia look like in the red. They kind of have, you can kind of see where the nucleus is, the little circle in the center. That's kind of like the brain that controls the cell. And then extending out from the microglia are all of these thin processes. So these kind of extend out in all directions, especially in a microglia in a healthy cell. So this image was taken from a brain that doesn't have Alzheimer's disease. So you can see the microglia, they're outstretched, they're looking for any like danger, anything they need to respond to in all those different directions. So then to kind of contrast that, we have an image of microglia in a brain with Alzheimer's disease. So I feel like here, if you look at the microglia, there's kind of that circular nucleus again, but there isn't any more like fine processes. It's a lot like more thick, a lot more angry looking to me, and it's a lot more in one direction too. So these microglia are responding to a plaque of some sort. They're really reaching out. They kind of get integrated with those plaques too, and they're just kind of, their whole purpose is all about like fighting off that, you know, attacking that plaque. So you can see there's quite a difference between our activated microglia, as we call them, or just the homeostatic or normal microglia. So that's a big thing that we look at, is we look at microglia and how they look in the brain at different times. We also, these images are confocal images. That's a special type of microscopy. So as you can see, we're looking at the plaque, and we also have microglia. There's also nuclei. It's just a little dim, so we can adjust that. But yeah, in real time, you can get these images pretty quickly and just scan like those regions of the brain.
Thank you for coming here. So this is the place that we're doing the immunohistochemistry, and this is the mouse brain. If you're scared, you don't have to look closer, but it's really small. And we make the specimen to put this mouse brain here and froze them to keep the structure or pathology intact. And as you can see here, this is the machine to cut the slices, and here is the sample. And yeah, this is the mouse brain.
So it's frozen. Have you put it in a medium?
Yeah, it is, which is called compound. So we want to cut the slice very, very tiny one, and now we are cutting 10 micron, and you can see this is the slices that we are using. Oh. It's really tiny, right? And we put these slices on that, and this is the one that we make the slices.
Yeah, so the protein that we study controls the function of the immune cells, called the microglia. And what our research shows is that different people have a different form of CD33. Most people have one form, the long form. Other people have the short form. And what our research is showing is that the two forms have a different impact on the microglia. The long form, which most of us have, is a brake. It stops the microglia from responding. The short form, that's only in 10% of the population, is like a gas pedal and it makes the immune system, immune cells better able to respond.
So what's the impact on Alzheimer's disease?
So first and foremost, it teaches us about the human genetics and why these two different forms correlate with Alzheimer's disease. So it's explaining what was known at a population level, but the implications for Alzheimer's disease, I think the most important findings is what we can do next with these findings, which is it shows how we could potentially develop a drug to make the microglia better to protect our brains.
Do you feel comfortable sharing some of the challenges you may have faced?
Sure. So many research challenges. I mean, there's tons of challenges. So many challenges that I wonder how we get it done sometimes. But we push ahead, and challenges for me are funding the research. First of all, second of all, finding the right people to do the research. You saw my team today. I have a wonderful team of scientists, so I'm quite fortunate right now. Like, for example, a year or two ago, we had a little turnover in the lab, and so I'm like, well, who's going to do the work? But we found some really amazing, recruiting some really amazing scientists.
Enjoyed it very much, and it was very informative, primarily, I guess, because I saw that many people were experiencing it. My husband did, in his last few years, experience Alzheimer's, and so I think that it's so worthwhile. I can see that certainly with all the work that's being done, that, they're trying to look at the different avenues and really how the, I didn't realize the influence of plaque. I didn't realize the influence of sugar. and Alzheimer's with, with diabetes. I mean, all these things are all interconnected, and so we've got so much to learn.
Honestly, I thought it was really eye-opening, something that I really enjoyed seeing how everything worked, all the research that was being done. So my grandfather, he suffered from Alzheimer's, and so that's something that obviously my grandmother's always been a big part of. She was really involved in that, so that's kind of been important to me, and something that I do care about just because it's affected people that I know, people that I love. Well, something that honestly, it was very interesting to me to see how much there was that we still didn't know. And there was still so much research that needed to be done.
My first thought was, why didn't I do this when I was 16 instead of doing electrical engineering? It's absolutely fascinating. And the depth that they're into and the fact that they're at cellular level, when they're looking at these things, tells me that it's only a matter of time before they can kind of crack the thing and do something for it, yeah.
My hope for the outcome of this research, I really hope that they're able to find like a cure, because like it hurts so many individuals, like people I love.